All, this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. Let's talk about another discussion today. This is a study from German, and this study also talks about antibody dependent cellular cytotoxicity, which can be put as one type of ADE. However, the cell that we are going to talk about today is the natural killer cell. We have not talked about natural killer cell in the context of SARS-CoV-2 and the vaccination since the beginning of our talks. So this is one of the very important talks because these cells are making their first debut in these discussions. Natural killer cells, I've discussed them before as well in immunology. They are the cells that are part of innate arm. They're part of innate arm. And their function is to kill the cells that are infected by a pathogen, or in this case, you would see that they are expressing the cells that are expressing antigens produced by the vaccine. So either way, the natural killer cells would be killing them. Now, normally, if you can just keep this much in your mind for a second, that normally natural killer cells directly interact with the cell's surface with their own receptors. I call them pervert cells because they touch every other cell and they kind of assess the cell's health by the presence of the surface proteins on the cell surface. For example, presence of my hair might tell my health. And if my hair just fell off all of a sudden tomorrow, then I may have a problem. So similarly, cell surfaces have cluster of designations or proteins on them. And natural killer cells can look at or sense the density of the protein from where they can sense if the cell is sick with cancer or maybe with infections. And they would then kill them, the cells, if they're sick. However, today's discussion and this study does not talk about this mechanism. Instead, they talk about antibodies dependent mechanism. A mechanism of natural killer cell killing our cells, but because of the presence of the antibodies. And these antibodies are generated in response to the vaccine or in response to the infection. So that is a discussion, fascinating discussion, and I'll let you make your own mind for how you would interpret that. And please tell me, what was your interpretation of this? So this is drbeen.com. Quick references. drbeen.com. In the description, there is a link to the drbeen.com premium account access, one-time lifetime fee. And that access is the fee is so less that I think you'll be surprised. Check it out. Okay, so here is the study. It is printed in or published in European Journal of Immunology, and the date is 13 April 2022, so hot off the press. This is another link which shows that CD107A is a marker of the natural killer cell activity. So if I can give you an idea, when the chickens, when they fight with each other, or the birds, when they fight with each other, they become all puffed up. Correct? Similarly, when natural killer cells are killing other cells, when they're active, they have a specific protein on their surface that appears. And that protein is called cluster of designation 107A. It's a cluster of proteins. So please keep in your mind that when natural killer cells are active, we can actually identify them if they are active or not by looking for 107A protein on their surface. That's a marker of their activity. If a natural killer cell does not have 107A on their surface, that means it's a resting natural killer cell. If they have 107A present on their surface, then they are an actively fighting and killing natural killer cell. This is another link. It is a rather, I believe, older study. So if you see here, this would be 2019 study. And this is not about the SARS-CoV-2. This is about dengue. But natural killer cells activated through antibody-dependent cell cytotoxicity and armed with degranulation, interferon gamma production suppress antibody-dependent enhancements of dengue. So this this mechanism can actually run the other way as well to suppress the ADE in case of dengue. But here you would see that it could actually be towards the ADE. And then this is a link that was in 2021, and it is printed in Frontiers of Immunology, 2 different antibody-dependent enhancement risks for SARS-CoV-2 antibodies. So with these references, now let's start our discussion with my drawings. So I think you will like the drawings today. And if you like the drawings today, please tell me that you did, because I spent a lot of time today to make them. So more possibilities of ADE within the innate arm. And I would also want to present to you today that this particular study actually proves that with vaccine, innate arm actually becomes more active than suppressed. And this happens with the infection as well. So here, the study is funded by German Center for Infectious Disease Research and Ministry of Health. It is a reviewed, accepted, and published study. Although the manuscript that we have is unedited and it may have grammatical errors and other edits fixed, but data and the tables and the conclusions and the messages will not change. It is printed on 13 April in European Journal of Immunology. Authors note, this is an important one, authors note that maybe this mechanism helps protect by activation of the natural killer cells to kill S1 or N expressing cells. So we don't actually know how bad it is or how good it is. These authors, for the first time, at least in my observation, this is the first time, maybe there are other studies that I do not know about, I am coming across the first study that is showing natural killer activation. So this is a huge study in that context, that they are finding NK cells activity during the vaccine, after the vaccine or the infection. And they think it may be protective. This could be the other way around as well. It's a hypothesis. So here, how does it work? So I'm going to present you the mechanism. The basic study, let me give you a summary. Let's see if I can do a 30-second summary. The summary is this. When a person is vaccinated or infected, their cells, regular common cells, will present the SARS-CoV-2 antigen on their surface, S1 protein antigens or nucleocapsid protein antigen or other parts of the virus, or if it is just a vaccine, then S1 or other parts of spike protein. Those cells that are presenting the antigen on their surface, those cells eventually are coated with antibodies. This is a normal, natural phenomena. The cell is expressing the antigens, immune system will become active, B cells will make antibodies, those antibodies will get coated onto these cells, and I would explain that a little later. Then the antibodies are used by the natural killer cells to bind with this cell and kill it. That is the basic summary of it. Killing of our cells occurs not only through the cytotoxic T cells, which is CD8 positive cells. Here, natural killer cells, the part of the innate arm, is doing this. That is the important part. And the cells are getting killed. Okay, so innate arms, natural killer cells, and this is the antibody-dependent cellular cytotoxicity. I should have written the word here, cellular cytotoxicity, that is, the cells are killed, or it is also called ADCC. The antibody-dependent enhancement can be further categorized as antibody-dependent cellular cytotoxicity, ADCC, that we're going to talk about today, antibody-mediated or dependent phagocytosis, and antibody-mediated complement-mediated cellular damage or complement activation. So these are 3 categories of ADE. So we are looking at one of the categories. So here, this is a natural killer cell. I always draw them as a cell with the eye patch. It's a badass cell. It's very, very active and very dangerous cell. Now, the natural killer cell has a few things to keep in our mind. Number one, they have a protein that they can release, and that protein will cause hole formation in the target cell. These proteins are called perforins. These are the same proteins that are present in the CD8 positive cells as well. Then the natural killer cells have another set of proteins in them, which are called granzymes. I always say granzymes are like grenades. Here, this little burning skull is the granzyme. Granzyme's function is that once a natural killer cell has punched a hole in the target cell that it wants to kill, then it would release granzymes that would go through that hole in that cell, the target cell, and then they would kind of detonate inside. And what is that detonation? They will go and activate the caspases. Caspases are proteins in the target cells, which when activated will cause the cell to commit a suicide or do apoptosis and die. So here, a perforin, here a granzyme. And here, if you see this little blue part, this is another protein on a natural killer cell through which it can bind with the antibodies. So you can call them, these are antibody receptors. This protein is named or designated as CD16, cluster of designation number 16. All these proteins are designated as various codes. But this is Fc gamma receptor as well. It's an antibody receptor. Good. So 4 things to keep in mind. I have not made the 4th thing over here, and that is CD107A, which is, I'll make it when I would show you that they are active cells. Perforins, granzymes, CD16. This is the setup. And here is a cell that is sick. Why is this cell sick? Because let's say we had a vaccine given to this person, and this is a cell, let's say in the deltoid, and the lipid nanoparticle went in there and the messenger RNA went in there, and the cell is now making antigens, and those antigens are going to be shredded and then they will be presented on the surface. Or maybe this cell got infected with SARS-CoV-2 and now on the surface there are antigens. So these little things on the surface are the antigens present on the surface. These may be S1 parts or part of nucleocapsid if it is an actual virus and so on. So this cell knows that I am expressing antigens on my surface. I am a target to be killed by cytotoxic T cell or by natural killer cell. Either directly, natural killer cell actually has a capability of catching or grabbing this cell directly and killing it, or through the antibody. So that is a problem, that antibody would actually enhance it. Okay, so now this cell, that cell that was expressing the proteins, on the surface of that cell, antibodies will come and attach with those expressed antigens. So imagine me for a second. I got sick. Imagine I'm a cell. I got sick. And on my surface, weird things appeared, which are parts of the broken antigens. Then my immune system is now active because there is either a vaccine or there is an infection. And so the B cells became active and they made antibodies. Those antibodies were circulating in the fluids and they reached me as well. And they got stuck on the antigens. Wherever the antigen was, with the antigen, the antibody became stuck. This is now this poor cell's setup to die. Usually what happens is that a phagocyte, for example, a macrophage or a dendritic cell, will pick up this cell through these antibodies and kill it. That is the antibody-mediated phagolysosomal activity or phagocytosis. Here we're going to look at natural killer cell jumping in the action because of antibodies present here. So take away from this diagram, antibodies coat this cell that is expressing antigens, foreign material on them, this process is called opsonization. Now what happens? Now what happens is that this cell that is expressing these antibodies is identified by the natural killer cell. Now, natural killer cells are running around everywhere in our body. They're part of the innate arm system. They are patrolling everywhere. So what will happen is, if you see here, the CD16 or FC gamma receptors on the natural killer cell will bind with the antibody. And now why are they called FC gamma receptor? So if I make a big antibody over here, so let's say this is an antibody. An antibody has two parts. There is a variable part which is changing, and it has a different binding system for various kind of antigens. And then there is a constant part. This part, which is constant across the antibodies, is called fraction constant. And then this part still have some variation, and that is the variation which makes them antibody type A or E or D or G or M. Remember when we say IgG, immunoglobulin G, in here the G means that the class of this constant part is G type. And these are only 5 types: G, M, A, and D, and E. Correct? So when there is a receptor that binds to immunoglobulin G constant part, then this receptor is called FC gamma receptor. What does it mean? Fraction constant of the IgG. Gamma is for IgG, G for gamma, G for IgG. Actually, IgG is called IgG because it is immunoglobulin gamma. And receptor is receptor. If we go back here, if you see the FC gamma receptor or CD16 has bound to the antibody. That antibody is bound to the sick cell. Good. Now we have a problem. Now this guy is going to become activated. And look at its head now. I have made a cluster of designation 107A. If you go back to my previous diagrams, you would see that that thing was missing because this natural killer cell was resting. Now it is not resting. It has become bound to a sick cell through the antibody dependent mechanism. And now this natural killer cell has become active, and it is demonstrating, it is showing 107A on its surface. So researchers used this marker to see if these cells are becoming active or not. So in their lab, they had the plasma from various patients or people, some infected and recovered, some vaccinated, some healthy, control, no vaccination, no infection. And then to their natural killer cells, they would present sick cells. And then they would look for their 107A marker to see if they become activated or not. And if they become activated, they would know that now this natural killer cell is going to start killing. Okay, so once this connection occurred, 107 is apparent, the natural killer cell has become active. Then what happens? Now this is the same natural killer cell. The drawing is a little different now. But if you see, what it is doing is, this is the hand of the natural killer cell. So in this case, I'm just drawing it. Normally what happens is natural killer cells release perforins in the local tissue fluids. Those perforins go in and they punch hole in the membrane of the bacteria or the virus or the cell, whatever is the target. This punching hole by perforins causes two problems for the target. Number one, the fluids from within this target cell or pathogen or bacteria will start spilling out. And now this is, you can say this is equal to cell bleeding. So cells don't have blood in them, but they have fluids in them. And those fluids are going to get out and the cell is going to die now. That's one. Secondly, through these holes, the enzymes that will be released would get into the cell and kill it. So here you are seeing that the perforins are released by the natural killer cell and they have caused holes in the target cell. Now remember, it is not doing it to every cell. It is the cell which is connected to this natural killer cell. So natural killer cell has on one hand grabbed this cell. If I give you a kind of a gory example, if somebody grabs another person and stabs them, this is what this guy is doing. So if it is not grabbing the cell, then it is not stabbing it. So normally they would connect with them first, and here the connection is antibody mediated, antibody dependent. That is the bad part of the antibody, or maybe the good part. Maybe it is protective, maybe it is not. And then, and to stay loyal to the paper, author said it may be protective. Okay. Now, next step, we have created holes in the surface of the cell. Then apoptosis is triggered by granzymes. Now this activated natural killer cell is producing granzymes that are entering this sick cell, which is now getting beaten up and punched hole in. And those granzymes are getting into the cell through those holes. And when they would get into the cell, they would now activate caspases, and caspases would tell the cell's machinery to activate that machine, the system of proteins that would kill the cell. So there are, remember, 3 steps so far. CD16 has to be engaged with the antibodies. So if there were no antibodies, this will not have been happening. Natural killer cell have direct capability of killing as well, but this paper is about the antibody-mediated enhancement, or ADCC. So CD16, CD107 became apparent, fins caused holes, and then granzymes are released that would go into the cell and kill it. Now what is the cell doing? This is the dead cell now. The cell has caused apoptosis and it has died. And here is the natural killer cell standing proudly saying, all right, I killed this one. Now, this is an interesting one. In the paper, in this paper, beautiful paper though, although small number of people, but again, the study is interesting. On the page 5, they have this little diagram, which is very interesting. This diagram shows, if you look at the lower part, it shows that here is a healthy cell. Here is this pink one is a natural killer cell. They got bound here through the antibody. If it is a healthy cell, there are no spike proteins or no antigens on its surface, so natural killer cell are just going to leave them alone. But if it is a SARS-CoV-2 infected cell, then they would kill, the natural killer cells would kill them. If it is vaccinated, and the researchers had used Pfizer-BioNTech vaccine cells, and if you look at below them, there is this little grading. ADCC in healthy, all empty, nothing. In infected, 2 grade, and in vaccinated, lot more, 3 grade. So what they're saying is that they observed this attack on the cell and killing of the cell by the natural killer cells in the case of vaccination more, and in the case of infection less. And then in the end, I want to read their abstract. I have found that this is the way to stay in line with their message. So even if my interpretation has caused any distortion in message, at least you can look at the right abstract and you can kind of see that, all right, Mubin may have missed a point here, but here is the summary from the authors. So this is the summary from them. COVID-19 caused by SARS-CoV-2 has emerged as a global pandemic, while immune responses of the adaptive immune system have been in focus for research. So they're saying that we all have been talking about adaptive immune system forever, the antibody production, the CD8 activation, the helper T cells. the role of NK cells in COVID-19 remains less well understood. And I remember in the beginning of the pandemic, there was a startup, I believe they were based in New York or Boston. And the CEO of that startup kept going on various TV shows and kept saying that natural killer cells are a very important part of combating this disease. We should give natural killer cells to people all the time. And we should make sure that the amount of natural killer cells are correct, and the people who are under stress and the adults have lesser volume of the natural killer cells, lesser number of natural killer cells. Kids have them more. Please make sure people get NK cells. Or I even did a video about how do we increase the number of NK cells by forest bathing and less stress and all that. And here we are 2 years later, that there is a study that says that NK cells take part. Now, are they harmful or helpful? Here again, authors, let's see what authors are saying. The role of NK cells in COVID-19 remains less well understood. Here we characterized NK cell mediated SARS-CoV-2 antibody dependent cellular cytotoxicity against SARS-CoV-2 spike 1, S1, and nucleocapsid NC protein. So they actually tested both the vaccinated folks' blood and infected folks. Serum samples from SARS-CoV-2 resolvers, here resolvers mean those who had the SARS-CoV-2 and have recovered, induced significant CD107A expression by NK cells in response to S1 and NC nucleocapsid, while serum samples from SARS-CoV-2 negative individuals did not. So this 107A expression means activation. So what you're saying here is that the presence of the or bringing natural killer cells near those cells that were from infected patients and were expressing the spike protein antigen or nucleocapsid antigen activated the NK cells. Furthermore, serum samples from individuals that received the BioNTech vaccine induced strong CD107A expression by NK cell that increased with the second vaccination and was significantly higher than observed in infected individuals. As expected, vaccine-induced response were only directed against S1 and not NC because the vaccine doesn't have nucleocapsid protein in it. S1 specific CD107A responses by NK cells were significantly correlated to NK cell mediated killing of S1 expressing cells. Interestingly, screening of serum samples collected prior to the COVID-19, this is a very interesting point. I think you would love this point. Interestingly, serum samples collected prior to the COVID-19 pandemic identified two individuals with cross-reactive antibodies against SARS-CoV-2 S1, which also induced degranulation of NK cells. So they took few people whose blood was present in the bank, and that blood was before the pandemic, and they did the same test on them as well. And two people's blood or the NK cells actually attacked the S1 protein. Now, why? They think it is because the normal coronaviruses also offer some cross-reactivity with the SARS-CoV-2. Taken together, these data demonstrate that antibody induced by SARS-CoV-2 infection and anti-SARS-CoV-2 vaccine can trigger significant NK cell mediated ADCC activity. I keep thinking of FLCCC, and identify some cross-reactive ADCC activity against SARS-CoV-2 by endemic virus specific antibodies. So here is one more very interesting, very important study. I believe that in the coming days, based on this study, you would see a lot more studies done, you would see a lot more pathologies connected, and you would see a lot more discussions to occur. So this is the discussion. Please do me a favor. We are at the end of it. So my usual messages: like, subscribe, and share. And Secondly, my apologies for yesterday's videos. Of course, it was the thing that I had mentioned before as well. I had my computer updated with the Windows updates a day before yesterday. And once again, it reactivated or enabled my devices, onboard devices. And this computer I bought from a very good place, and it was a high-end computer, but it's such a crappy onboard network card that when that becomes active and takes over, it has a hugely bad network performance. So I disabled it, and once again, I have an external network card attached to it, or the dongle, and I hope today it was fine. So with this, please do me a favor. Look at the description. There are some links. One is for Dr. Bean's premium account, really low price. I think you would like it. And we would change the price after some weeks or months. But at this time, you can take advantage of it. And this is only for YouTube. And then there are links. If you would like to support this work, become a patron, $5 a month, or become a Substack member, and $7 a month. And you would be able to support this work that we're doing. Or you can use PayPal, or you can use Buy Me a Coffee as well. And if you didn't want to, that is totally fine as well. With this, thank you very much. I would see you in the Chit Chat channel, which is, if you wanted to search Cool Beans Cafe Live on YouTube, if you search it, we will be there doing a quick chit chat. Thank you. See you soon.